reproduce by means of flowers. Flowers are showy, colorful structures made of modified leaves. A flower usually contains both male and female reproductive structures. A flower can have up to 4 types of modified leaves: petals, sepals, carpels, which consist of a stigma, style, and ovary, and stamens, which consist of an anther and filament. Ovules and megasporocytes are present inside the ovary. Let's review the sequential stages of the angiosperm life cycle. The process that occurs in the ovary, which initiates the development of female gametophytes, is the division of a megasporocyte by meiosis. Inside an ovule, the megasporocyte, 2N, undergoes meiosis to form 4 cells, but only one of these cells survives as the megaspore, N. The nucleus of the megaspore divides by mitosis 3 times, resulting in one large cell with 8 haploid nuclei. Membranes then grow between these nuclei to form a multicellular structure called the embryo sac, which is the female gametophyte. The large egg cell is located at one end of the embryo sac. A diploid cell within the anther divides by meiosis to form 4 microspores, N. Each microspore, N, divides once by mitosis to form 2 cells. A tough wall is formed around the outside, creating a pollen grain, which contains a male gametophyte. Pollen grains are transferred by wind, water, or an animal to a sticky stigma in a flower of the same plant or another individual of the same species. After a pollen grain lands on the stigma, it forms a pollen tube that elongates downward through the style into the ovary. 2 sperm nuclei, N, form inside the pollen tube. The pollen tube enters the ovary and releases the 2 sperm nuclei into the embryo sac. One sperm nucleus fertilizes the egg, forming a diploid zygote. The other sperm nucleus fuses with 2 nuclei in the center of the ovule to form a triploid 3N cell that will develop into a nutritive tissue called endosperm. This process is called double fertilization. The triploid cell divides rapidly by mitosis to form endosperm, a tissue rich in starch, lipids, and proteins. The zygote divides by mitosis to form a sporophyte embryo with a rudimentary root and 1 or 2 cotyledons, seed leaves. The integuments of the ovule become a tough seed coat with the embryo and endosperm inside. As the ovules develop into seeds, the ovary surrounding the ovules develops into the fruit, which will protect and help disperse the seeds. The fruit falls to the ground and seeds germinate. A seedling uses the food stored in the endosperm and cotyledons to grow until it can begin photosynthesis. The young plant grows into a mature sporophyte that produces flowers, and the cycle continues.